scientists have rapidly restored movement in 2 paralyzed monkeys in an operation which could soon be used to help people with damaged spines. The monkeys walk normally again after having the pioneering device fitted after just 6 days. Because humans have very similar anatomy, scientists are confident that the same result is reproducible in humans by the end of the decade. The researchers used an implant in the motor cortex of the brain, which controls the movement of limbs. Amazingly, once the device was implanted, it worked in the monkey without any training to help them to use it. The neural interface device, which is under the monkey's control, bridges the spinal cord injury in real time and wirelessly. The system decodes activity from the brain's motor cortex and then relays this information to a system of electrodes located over the surface of the lumbar spinal cord. Below the injury, electrical stimulation of a few volts delivered at precise locations in the spinal cord modulates distinct networks of neurons that can activate specific muscles in the legs. This pattern emulates the way a monkey would walk normally. Previous research has shown similar implants can restore planning and movement in the control of robotic hands and in one case, a patient's paralyzed hand. But the way leg muscles work is much more complex and so a bigger challenge. But as the surgery used components that have been approved for research in humans, the technology could be rapidly developed for human use. The scale of disability globally caused by spinal cord paralysis is huge, between 250,000 and 500,000 people each year according to the World Health Organization. Around 37% are caused by collisions with cars, 41% caused by falls and 12% due to sport. Gregoire Courtine and colleagues tested the device in 2 rhesus monkeys whose legs had been paralyzed by a partial cutting of their spinal cords. After being fitted with the devices, one of the monkeys regained some use of its paralyzed leg within the first week after injury without training, both on a treadmill and on the ground. The other monkey took 2 weeks to make a similar recovery. The research reported in Nature Communications said. Professor Courtine of the Swiss Federal Institute of Technology in Lausanne said, "This is the first time neurotechnology restores locomotion in primates, but there are many challenges ahead and it may take several years before all the components of this intervention can be tested in people." He added, "To implement the brain-spine interface we developed an implantable, wireless system that operates in real time and enabled the primate to behave freely without the constraint of tethered electronics. We understood how to extract brain signals that encode flexion and extension movements of the leg with a mathematical algorithm. We then linked the decoded signals to the stimulation of specific hotspots in the spinal cord that induced the walking movement." Dr. Mark Bacon, executive and scientific director at Spinal Research said, "Paralyzed patients want to be able to regain real control, that is voluntary control of lost functions like walking, and the use of implantable devices may be one way of achieving this. But the technology solutions need to be powerful enough to manage natural movement under voluntary control in real time whilst not being cumbersome or intrusive. That's what will take it out of the lab and into the clinic." The current work is a clear demonstration that there is progress being made in the right direction. The fact that the components of the brain-spine interface are already approved for application in humans makes the translational pathway more straightforward, and a proof-concept study in patients may not be that far away. Andrew Jackson of Newcastle University, in a commentary, added that given the rapid development of other similar neural devices from monkeys to humans in recent years, it is not unreasonable to speculate that we could see the first clinical demonstrations of interfaces between the brain and spinal cord by the end of the decade. The research was carried out in China, but according to EU-approved standards for a laboratory, the researchers said that monkeys were used rather than rodents, as they are very like humans in the way their brains activate during walking, how they recover from injury, and the technology that is used would also be similar. Dr. Jackson said that it was notable the research was carried out in China. He said, "The use of monkeys for neuroscience experiments continues to be questioned in the media, and animal rights groups are making concerted efforts to ensure that restrictions on such work are tightened in both the United States and Europe. Many scientists may lack time, energy or resources to travel to China," he said. "The danger would be that there is thus a real danger that the development of treatments for debilitating neurological conditions will be delayed if high-quality, well-regulated research in monkeys cannot be performed in Europe and America owing to increasingly tight regulations." Professor Simone Di Giovanni, chair in restorative neuroscience at Imperial College London, who was not involved in the research, said the research was very promising and exciting in an area of high priority. He said it was an important step forward in our understanding of how we could improve motor recovery in patients affected by spinal cord injury by using brain-spinal interface approaches. Before used in humans, more studies in larger numbers of animals would be necessary.